ecology. So now he received this well-deserved award. So congratulations to Pablo for.
Congratulations, Pablo.
Yes. So the emergence of ecology through metabolic cooperation and recursive organization is the title of Pablo. No?
Pablo, you are mute.
Yes. Is self-censorship.
Yeah, thank you. Oh, thank you. I was saying thank you, friend, and I was saying also that I really like this title. Probably is the best part of the talk. So we'll leave it there because of the implications. You know, it's like all the words came together, and I will try to provide try to put this in context and see if we can talk a little bit about this metabolic cooperation and recursive organization, which is actually a project. The name of the project we have with Rolando and Mauricio and Cristobal, and also where Leonardo is involved. So I'm just going to try to give you some conceptual framework and some results of the model where I see them, how they fit into this framework. So one of the things I'm going to emphasize is that metabolic cooperation somehow implies two things, and that I will say that there are kind of a general principles in ecology. One is the one thing is associated with the niche construction, or the way organisms change their environment, and by changing their environment, they change themselves, and also implies that eventually organisms end up being energetically equivalent and also equivalent in fitness. So that's are kind of the major things I'm going to try to sustain during the talk. So metabolism has been important. It's core to all the theories of the origin of life that we might think of, from Maturana Varela to Tibor Ganti to Morovitz, and also to Eigen and the hypercycle. I mean, we have cooperation as an emergent phenomenon that classify or.
Excuse me, Pablo, can you put a
larger screen?
Larger screen.
Oh, there you go.
Yes,
That's
okay.
better.
Okay, perfect. Okay, so so you have that metabolism is a kind of a quintessential to understanding the origin of life, and is kind of important in all the theories that we might think of, and life itself is explained as the emergence of a metabolism, a network of metabolic interactions. So this is fundamental. I would tell and say that there are some general principles associated to this, and I will just talk about two of them, which is the niche construction, and the other is the niche equivalence, or also implying that there are some fundamental symmetries in organisms that are a result of ecological dynamics and this cooperation. So let me give you an example of this niche construction through metabolic cooperation, and also how this implies equivalence among organisms in terms of their fitness. And this is the simple example is microbes in a kind of a long-term cultivation in this container. Okay, so there are basically three ways you can culture microorganisms. One is using a chemostat, where you have an inflow of resources and an outflow, where the other is a serial transfer, where you put the microorganisms here, they grow up to stationary phase, and then you kind of move a sample from there to the next fresh media, and you keep the thing going. And these are long-term experiments in evolution that you can do here, and there are some things that has been running for years already, doing experimental evolution. So, but the other one is the long term. The long term was something that Roberto Golter, a Guatemalan in Harvard, came up with. He said, well, most of bacteria in real life, they live kind of stressed, you know, looking for food or kind of interacting in doing the long term in not very kind of these optimal conditions with influx of resources. So he left this bacteria in a long-term experiment. So he just actually what he did was to just seed this container with an isogenic meaning all the individuals are the same genotype, pretty much isogenic strain of E. coli in this container, and he looked at what happened with diversity and resource competition and so on. So, but he didn't add anything. So what you expect is that under this prolonged extravasation, when you seed the system with millions of copies of this isogenic strain, here CFU is colony forming units. It's a way of measuring the number of bacteria. This is time. So you see the typical dynamics, you know, things, the bacteria stay in a lag phase, like trying to understand its environment and put into run all its metabolic activity, and then it starts growing exponentially when it's kind of is ready. And then when the food runs out, it's get into this stationary phase, and then it will start to decrease. But in this experiment, and this is the kind of a surprising thing, the decrease took a little bit, which is, and look at this. This is the time frame is in month. This is hours here, and then you have month and month. So eventually, until 60 months, meaning 5 years, you still have the bacteria. And remember, you put an isogenic strain here. Everyone was identical. And you leave it here without, only with the initial food. And then you have the thing grows, start dying, but it keeps kind of a diminishing in abundance, which is fine, but also it keeps going in the long term. What is called, this is the long-term stationary phase. What happened here is that in this area, I'm going to zoom in a little bit here. What happened is that there is, during this long-term stationary phase, there is a periodic selection regime, meaning that the original strains gets transformed to this strain in red, and then there is a blue one that shows up, and then this purple one, and so on and so forth, until you have, after 3 months, you have this coexistence of strains in this kind of confined system. So somehow you have the emergence of an ecology here, and we wanted to understand this, what was happening with this model. So with this system, and we did a little model a while ago already on how this diversity can emerge, and how you go from this competitive exclusion to this neutral coexistence in these bacterial ecosystems, which we think is a toy model for eventually real complex ecosystem with microorganisms. So the model is pretty simple. So you just follow up the phi here is biomass. So you use proportional biomass in the container, and R is actually the growth rate, which is associated with W, which is the quality of the habitat where the bacteria is, and give you the quality affect the fecundity minus the mortality, and the quality depends on the supplies minus the consumption of resources, and the supply is a function of this lambda supply rate, which the initial condition is what you put, but then lambda is somehow taken over by the bacteria, because the bacteria becomes the ones that provide the resources for themselves, and I'm gonna try to explain that. So supply is a function also of this W. If W is top quality, then you don't need to supply anymore, and this goes to 0. And consumption is a function of fecundity, how you transform the environment into your own biomass with an efficiency epsilon here. So with this equation, you can compute equilibrium, and after doing some dimensionalization, you can just basically dividing, expressing all this in terms of 1/f time, and so it's fecundity time. You end up with a kind of expression where you can do the adaptive dynamics for the system, and you can compute the same things that Nikolai was showing. You can compute the fitness exponent for the a new mutant in the populations to see what would be its fate, and also you can compute the fitness landscape or the fitness gradient of the system. So basically, what we have here is that you realize here that this is always minus less than 1, so this is negative. So that you have an evolution of the trait, where the trait is this W star, which is the relationship between mortality and fecundity, and you have that the evolution goes to the left here, the strategy evolution, and mutants to the left will always kind of invade because they have higher fitness. Okay, and the interesting thing is there is a point where the fitness becomes flat, and that's the point of coexistence. So somehow you have periodic selection to arrive to eventually a system where things coexist and have equal fitness. That's pretty surprising. We understand the model, but we don't understand the biology now, because what is going on here? So the hypothesis is that the importance of this phenomenon, which is called cross-fitting, which makes it's very well documented, and basically is that you have strains that can take a particular metabolite and actually generate other metabolites as part of their metabolism, which can be used by different strains. So they are coupled in terms of these exchanges of chemical species. So basically, these things are coupled because they need each. There will be this one here and this one here need the existence of this guy CV103, because this guy's the CV103 strain can take a particular metabolite from the environment and make this to become a resource for these other two guys. Okay, so we hypothesize that this is what is going on here. You have an original strain that eventually generate, as a consequence of its metabolite, it transformed the environment in copies of themselves, but also produce another kind of a metabolite, which can be used by a different strain, which in turn produce a different kind of metabolite as a consequence of its metabolic activity, and so forth and so on. So you have periodic selection, because eventually you run out of the food and you transform it for someone else. And eventually you have to, if you close this loop here, you will go into a phase of coexistence, you know. And so we did a particular toy model. I'm not going to give you too much details, but you have a strain. It has a probability of surviving. It can go extinct too, or death, and you have you have a probability of reproducing and mutation. Also, depending on the number of resources on the environment, and then we are pretty much working on this right now. So I'm gonna just give you a sense of the dynamics that we find, and this is the simulations. Is you have the abundance of the genotypes, of the different genotypes in the population. You see this, there is a periodic selection where they replace each other, and eventually there is a point where there is this coexistence in the population. And interestingly, the metabolites abundance was fluctuating a lot, and then eventually it becomes much more stable. So we are trying to understand this as we speak, actually, and you have also the number of phenotypes in the population. There is this kind of a transition here at some point. What we think is basically the integration of these metabolites that generate this big graph of interactions where each one depends on each other. And that's why you have coexistence and the emergence of this dynamics, which is pretty much like neutrality, so to speak. So why I talk about niche construction here? Because you have the regional strain. The regional strain, what it does is transform its environment into a different one because of its metabolic activity. So it makes copies of themselves, transform the environment, generates a waste, quote unquote. But since there is no internal, external addition of resources, this transformed environment becomes a resource for someone else that it might be himself after mutation. So this strain mutates, generate a different strain that changed the environment, and that environment also affects its own evolution. So you have this recursive evolution through this kind of niche construction that somehow for organism in this confined environment, if you create this big loop, you eventually will generate conditions for coexistence, and where everyone becomes kind of a cooperative and they coexist. So this is basically the picture here. You have individuals and you have environments. So you have natural selection going this way, affecting the individual fates, but niche construction goes the other way in terms of affecting the environment. So you have this kind of interesting dynamics. So it seems that metabolic cooperation then lies at the basis of the emergence of the simple ecologies, as I show you for the bacterial case. And also, since these entities are kind of dependent on their metabolic transformation chains, there is room for the emergence of some fundamental symmetries in terms of fitness. So let me elaborate a little bit on this and show you a principle of niche equivalence, which also capitalizes on metabolism. And it is well known that the metabolism, which is the addition of all the chemical reactions going on in a particular entity, all this activity increases with the size of the system, of the biological system. And this biological system, I mean organisms, and the way of assessing the size of the system is through the mass of the organism. And they follow these very simple relationships, which we call scaling relationships, and which are very interesting because it tells you that there's some fundamental symmetry among all these things, because an elephant could be it's just an amoeba blown up, in terms of they are all obeying the same rule somehow. So there's this fundamental symmetry in this. How was this explained? One of the hypotheses is that this dependency between the size and the metabolic rate is associated with the way that networks transform and actually transport resources within individuals. And those networks have this particular kind of fractal branching. And if you assume this and you pump resources through these networks, eventually you can compute that the metabolic or the flow will be proportional to the size of the system raised to a particular exponent that tends to be basically 3/4. Okay. There was the explanation came out in a paper in Science back in 1997. It was heavily criticized, but still there have been a lot of new additions to this modifications. But I think it's holding out pretty well, and it gives rise to several disciplinary innovations in ecology, and particularly the metabolic theory of ecology, and which is kind of a very trending issue. But one of the things, we have been working with it for a long time, by the way, since the 1990. We started working on this with Sergio, actually, and Sergio Navarrete, who is speaking after me. And one of the intriguing things was that lies at the core of this metabolic ecology is this other relationship. which is the Moore-Damuth plot. You know, suppose, this is very interesting because this is another regularity. Suppose you go to the world and you sample a mammal. In this case, suppose all these are herbivorous mammals. You go to and sample mammals in their habitat. You know, you go to a savanna, hardcore savanna, and sample mammals there. You go to the Mediterranean, you go so on and so forth, and then you take the samples and you plot the abundance, the numbers per square kilometer versus their size. And cha-chan, you get this. So it's like, you know, this is very interesting because it's associated with the Red Queen and all that. And, but it's very interesting because do you know what the slope is? So the slope is -3/4. So it's look like if your individual metabolism is 3/4 and your density is -3/4, there is a fundamental symmetry that tells you that the maximum number of individuals that you can have will be actually the number of resources over your metabolic rate. So this maximum number will be, the prediction is that will be proportional to mass raised to -3/4, which is actually what you find. But this assumes that all individuals use the same amount of energy. I mean, all populations, because if density runs through mass through to the -3/4 and a metabolic rate is mass to the 3/4 but positive, then the aggregated use of energy of a population will be D times B, which will be independence of size, meaning that things will, every population in a particular ecosystem will control the same amount of energy. This is a plot of the maximum population size from phytoplankton viruses up to sequoias and elephants, and the slope is -3/4. And that implies that the energetic equivalence rule, which is what I just told you, is the population energy use will be the individual metabolism times the number of individuals, which will be independent of size. So everyone, regarding to size as a trait, everyone's used the same amount of energy in a particular ecosystem. So that was the thing. So back in the 90s, this was just proposed, and so we decided to see if this actually happened within a real ecosystem, because this was made out of compilation of particular populations in the world, maybe not necessarily in the same ecosystem. So we did this back in 1990 with Sergio and Juan Carlos, and we took advantage of this intertidal system where there has been an experiment where humans were excluded from this rocky shore, and the exclusion of these humans led to a completely different landscape. And as I will show you here, this is the marina station as it was back in the 90s or 80s. Now it's much different. It's a lovely place. It's a perfect place to spend a sabbatical if you want. The new. Let me just a kind of a little bit of advertising here. There is now 2 blocks here going out into the sea, so you can work in a wonderful environment. And everyone speaks French in Las Cruces. That would be wonderful. But this is the kind of people, the kind of fishermen that collects sea urchin and loco, which is a delicious, very well regarded food item, and all these fishes from the rocks and limpets. So if you take humans out of the system, you have a complete different picture. When humans are present, you have this particular physiognomy, where humans are excluded, change the abundance of the organisms, and yet the relationship between density and mass stays the same. So this was kind of a very interesting, because if you perturb the system, these points move around here in this space, but they somehow keep satisfying the same relationship. And later on, it was also documented for other kind of marine ecosystems and also forest, where after hurricanes, they rearrange, they change the density, but they satisfy the same principle. So it's kind of interesting to see that. So what this implies is that somehow species also achieve this symmetry in terms of energy. And Lee Van Valen, a long time ago, said fitness is the control of energy by organism, because it's the energy what you use to make copies of yourself and reproduce. So somehow this implies a kind of a neutrality. And now neutral theory, you know, has become very popular in this since 2000 after in ecology, particularly after Steve Hubble published his book on the pompous title, The Unified Neutral Theory of Biodiversity and Biogeography. We showed recently with Rolando and Mauricio that the unification actually is not right, because what the biogeographic model under this master equation here is not compatible with this. So, but anyhow, they propose this master equation, and if you assume that all the species are equal in terms of their demographic per capita rates of birth and death, you can show that this converges to the log series distribution. And that's what's the major kind of accomplishment of Hubble, was to show that, in fact, the predicted invariant density for this was this log series. But actually, as happens very often in science, this was already derived back in 1978. So because someone else did all the work and did the master equation approach to Fisher log series. So, but anyhow, it was a nice result. But it's interesting that that you actually using neutrality, you can predict this, and also neutrality means that individuals are equal in terms of their fitnesses. So, which is what I've been talking in the first part of this talk. So if they are equal, then we wanted to explore this and see maybe there is a more fundamental kind of process here that might applies to different biological entities. So, but we didn't want to take this route of using master equations because there was a problem there, but we took a different, more traditional route, which is use the diffusion process, which has a very distinguished history in biology associated to Seward Wright, to Ronald Fisher, and Andrei Kolmogorov, who actually provided basis back in the 30s and told these guys how to use what they call it the partial differential equation for evolution. So basically, what Seward Wright derived was if you have a specially structured population where you have a given frequency of an allele in the general population, you can predict basically the density for the probability density for the frequency of a particular allele in the subpopulation here, using deriving this, which is a beta distribution. So he said that gene frequencies follow a beta, and it's being kind of interesting that genes follow this distribution. And so we did this also with Rolando, and some results he derived back in the 70s to show that, in fact, under some assumptions in terms of the type of birth and death rates, we can derive a beta distribution for abundance too. Basically, when J is the number of individuals in the population, and when this tends to infinity, the process converges to this stochastic differential equation here, this diffusion that in the time limit converges to an invariant distribution. This rho here, which is a beta distribution, assuming that different individuals do not, different species do not interact, and also that the birth and death rate follow this particular kind of a structure here, where C is everything that affects death or birth rate, which is actually affecting the fluctuation or affecting fluctuations in birth and death because of external influences or things that might be going on within our system that we don't know. And that's why the C ends up here in the coefficient of the Brownian. So it was published 2017, and we show that when you express abundance in terms of frequency, it fits as well or better than the neutral model log series distribution. So first step, we show that, but maybe this assumption of equal fitness that I've shown you, there is very interesting way of supporting in ecological system might be much more fundamental. So we just went beyond the analogy with genes and the frequency of gene within populations. Now we have the frequency of individuals within the species. And the next step moving up is to see if these neutral assumptions using diffusion approximation might actually also explain why we have a particular proportion of a species in a particular place. And this will be the richness, the species richness distribution. And this is actually the proportional species richness distribution, which is basically, suppose you go to, you sample all the islands in the world for mammals. You look for mammals in the islands, you know, and suppose now that the assumption is, your assumption will be that all places are kind of similar, which we know they are not, but let's assume that, then we predict that that will be the distribution for this particular bird species and mammal species, and you can see they fit very well the beta distribution. But we can go somewhere else too. And what if we analyze now the abundance of gene families within genomes? Because the process of genes and individuals are similar. I mean, you also have a birth death process. You have gene duplication, mutation, all this sort of processes that are akin also to migration, as Nicola was saying. Use all this lateral gene transfer plasmid, and also you have gene extinction. So we did that with Evandro, and here is for 30 million genes in bacterial proteomes, and also eukaryotic and others, and the fit to the beta distribution is very remarkable, and that's work in progress. And so just to summarize, the point here is that there are ways by which we can understand the emergence of neutrality as a byproduct of some very essential, at least in bacterial cultures, metabolic cooperation. Somehow, all ecological systems are somehow based on this, or do have this kind of an interaction in terms of one organism producing environment for other. We call it ecological engineering. But also you have that organisms decay, they become resources for others, even when you die. So we are coupled to the rest of the communities with which we interact. And part of this coupling, if we take into account all the metabolic activity of organisms and the density they achieve, suggests that this gives rise to some fundamental symmetry, you know, that is reflected in equal fitness across the species, which, if we use that analogy into models, we can actually see that it might provide a good fit to the dynamics we observe at the level of genes, at the level of individuals, and at the level of species. So we are working on this in formalizing all these approaches right now, and thank you very much for your attention. But one of the puzzle things we need to know is the mechanism of this fitness equivalence. So with this in mind, I will finish right here. 35 minutes. Perfect.
Thank you. Thank you. Thank you very much,
Pablo.
So we have time for some questions or comments.
Maybe I could ask a question.
Yes.
Do you hear me? Yeah.
Yes.
I was wondering about, the allometric relations you gave. So in terms of metabolisms and species abundance or maximal size or things like that. Do you know if there are models trying to make this emerge? I mean, in a sense, consumption models of consumption of energy or things like that.
Right.
That could allow to explain that or to understand.
Yeah, there have been a couple of attempts. I would say some of them has been more successful than others, but basically using a food web. For example, I guess Simon Levine and Joshua Bites did something back in the early 2000s using some food web models to actually see how these slopes could this emergent kind of allometries can show up. I would say that I always had the flavor that there might be there have to include some. They may have to make some assumptions to begin with, in terms of individual metabolism. So if you put metabolism with a particular exponent, then you somehow things are not. It looks like a little bit of tautological thinking, but yes, I mean, but more kind of interesting models I haven't seen. There is a model that try to explain why you have the 3/4 for metabolism. Okay, there's plenty of those, and there is a big discussion because they're the 2/3 crowd, and then there is the 3/4 crowd. And everyone saying, "Oh, your theory is wrong because I found this relationship that is closer to 2/3." Say, well, yeah, who cares? I mean, just make a good theory, and we'll see from there. But there's been a lot of discussion, but there's still something that I would love to see more theory being developed in terms of explanatory for that. But still, yeah.
Thank you very much.
Thank you, Nicola.
I have another one, but maybe I can wait. No, thank you again for the very nice talk. It's quite intriguing, all these strange relations or common features. I was wondering about the beginning of your talk where you were explaining the experiment with bacteria in the environment, in the fixed environment. And I was remembering about, maybe for sure you know about this experiment of Lenski,
right?
Who made a culture for decades of the same bacteria. And do they observe similar? So you have this strange diversification thing that occurs after a while. And
right.
So do they observe similar things? Do you know?
No, yeah, I mean, that's the main difference, that they do the series transfer. And so they have this serial transfer supposed at different temperatures. And so the bacteria is always kind of is a prisoner in a 4-star hotel. It's always kind of transferred to a fish media and grows again. And so, but since this is carried out in different chambers with different temperature or different quality of resources, yeah, there is evolution to different kind of but it's more kind of a directed evolution towards diversification from the initial conditions. Here is real life. You know, it's like you have to solve a problem because you run out of food. We actually use the same model to understand human population dynamics in a finite planet. You know, which is we diversify, we generate technology, and we eventually capitalize, but we affect our resource base too. And yeah, here, but here is much more different because you really have a struggle for resources. Yeah.
Thank you.
Pablo, Clara has put a question in the chat.
Aha. Let me see. Clara, in the long-term microbial model, is there a limited number of mutants that can coexist, or it varies a lot per cycle? Well, we haven't seen that there might be, nobody has talked about the limitation to the mutants number. It looks like, because, well, for one thing, is that nobody has, that I'm aware of, done the genomics on this. That's something that we wanted to do, is to do a more kind of sophisticated analysis. What Coulter did was just observing different colony morphotypes and do some very kind of a genetic profiling, but very general, to show that there were different strains. But it is something that it needs to be done, is to show actually the diversity. So part of the project we have is to do this the next year, and also do the gene annotations, so we can actually see the interaction among the organisms in terms of what they are producing and for others, you know, so we can actually see if this metabolic cycle really emerges or not, using the genetic profiling of the organisms, the genomics. Yeah. You're welcome, Clara.
So, no more questions? Well,
yeah, can I make a final comment? Is that the issue about fitness is that because it's been very kind of controversial, that claiming that everyone has the same fitness or everyone is equal in their per capita birth and death rates, and the point here is not that everyone has the same fitness, is that end up with the same fitness because they are trade-off. You know, there are trade-off between competitive ability and dispersal. There are trade-off in terms also of competition, but also there are trade-off in cooperation in terms, or more than trade-off. There is some fundamental integration of information in this metabolic network, where you index your life to someone else in the network, because you depend on that other to be providing you with food or resources. So when that happens, everyone's get integrated, and you become a cycle which might be sustainable. And a lot of people now is talking about persistent selection, is that some of these cycles may be more persistent than others. So it's a very fascinating subject. So thank you very much for hearing me, and thank you, Rolando and Leonardo, for organizing this nice workshop.
okay, so
Thank you.